So today we're going to talk about non-Mendelian genetics. So far we have been doing Mendelian genetics where we talk about those three laws, and that's a normal Punnett square. And this may look similar to a normal Punnett square, but when we talk about what these genotypes do, I think you'll see that there's a pretty big difference between the two. It's not overly complicated. It's just another little tweak of what actually happens. Science rarely has absolutes. And this is one of those where we talk about Punnett squares. Typically, we're talking about Mendelian genetics, but today is going to be non-Mendelian genetics, the stuff that Greg Mendel did not address really with the pea plants experiments that we've talked about so much. So there's two things. There's incomplete dominance and codominance. It's going to be a pretty quick day of notes. Let's get started. So incomplete dominance means, if you think about what these words mean, we've got dominance, and this is going to be not complete. So there is a dominant letter. We see capital letters here, capital B's, big B's, but it's not going to be the same dominance as we would typically see with Mendelian genetics. So let's say that if there's three colors, you can talk about fish color or hair, feathers on a bird or whatever. It doesn't really matter. We're just going to say that blue is going to be that dominant gene. If that dominant gene shows through, it's going to be blue. If the recessive gene shows through, it's going to be red. So in this example, we've got two blue organisms, big B, homozygous dominant, with a homozygous recessive, little b, that is red. You can see I indicated that by the colors that I wrote them in. You would expect, now this is the Punnett square though, you fill out the Punnett square very similarly. However, a big difference is what these genotypes actually mean. So you fill out the Punnett square, the genotypes are the same as normal. You bring the big B down, little b over. So this was going to be heterozygous, just like all the other boxes. The difference is what this actually means. These genotypes will actually not make a blue skinned or feathered, whatever it is, organism like we would normally see. We would normally say this dominant gene is making this recessive gene shut up and be quiet, that it's not able to talk and show itself. However, in this incomplete dominance, when we're talking about incomplete dominance, the phenotype is actually blending together. We've got a blue and a red, and we'll just say in this case that those two genes together make a purple. And now if any of these two, if we were to do a two-step, if any of these two were to reproduce, their genotypes, big B, little b, if they breed together, or if it was self-fertilizing or something of that nature, you would end up with, again, fill out the Punnett square like normal, but this genotype is going to make that organism blue, and this genotype, like we talked about earlier over here, is going to make that organism red. And then there are 50% chance of heterozygous organisms. So it's not crazy different. There's just going to be a mixture. It is not a complete dominance. It's incomplete dominance. The phenotypes are blending together. Instead of there being one or the other, it is a blending together. So even though we're talking about two traits, we actually have three different options here. We have a dominant trait phenotype, a recessive trait showing off in the phenotype, or heterozygous is going to be a mixture of the two. So if you think of incomplete dominance, you can think of like it's an in-between. It's a blending together. It's not one or the other. It's incomplete dominance. And then we get down here to codominance. So think about co-show. Codominance is going to show both phenotypes. And so you might imagine a cow that is white with black spots. Or it's got two colors. It's not a mixture. It is both of them showing at the same time. So in this example, we're going to use a red flower with a white flower. And obviously, if I have a white board and I put white marker on there, you're not going to see it, so I just replace that with black. Both of these are dominant traits. We've got a capital. When you put them together, you get a little bit of both. You see right here, this is a red flower. Right here, this is a white flower, or in this case, black flower. When you mix them together, both are capital, which is a little bit odd, but both of them are going to be capital, and they're going to be some red petals and some white petals, or how I drew it, black petals. You can see the legend of that here, the key to that here. Homozygous dominant W's is going to be white. Homozygous dominant red, or ours is going to be red. And then if there's a mixture, it's going to be both. It's going to be a flower that has a little bit of red and a little bit of white. And another way you will likely see this is with blood typing. So blood typing, there are two dominant genes. There's an A, an A allele and a B allele. These genes together, if you have both of them, then you have AB type blood. And if you've never heard of this, your blood type, it's a huge deal. So you have A type, you have B, you have AB, or you have O. And there's different perks to having different ones. Some people can donate blood and anybody can receive it. Some people can receive any kind of blood. Some is more rare, some is more common. But you can look that up on your own. There's plenty of cool videos about that. I want to pay attention to what these black letters are. So if there's an A, so this is the mom. If the mom donates the A allele and the dad donates the B allele, that child would have AB blood. I wrote that in purple. If the dad donates this recessive, this O, and the mom donates the A, it'd be big A, little o. That's going to be A-type blood. And then there's B-type blood. Same thing, if there's the B and the O, that's going to be B-type blood. And let me stop right there. Both of these, another way that you could have A-type is if it was two big A's. Big A, big A. That would be A-type. And big B would also be big type B. The only way you get O-type blood, because it's a recessive trait, like we go back to think about Mendelian genetics, the only way that you're going to show that recessive trait completely is if it's all by itself. So you have O-type blood is a recessive type, blood type. And you can get into A plus and A negative and stuff like that, but that's for another day. You can go look up that on your own if you'd like to. This is a simple way of showing this. This over here is the exact same Punnett square, except it uses these eyes. So these eyes are there to show you that these two traits, A and B, are dominant, and O is recessive. And so again, big I with a little superscript like an exponent up there as an A up there as an exponent is going to be A, and this one's going to be just like B, and this lowercase is going to be like the O's. So you'd fill this out, it would be filled out the same, but I just want you to understand this is the easier way of writing it so you don't have another letter to draw or write, but the Punnett square would end up, you would have 25% AB, 25% A, 25% B, 25% O. And so these are the same parents on the outside. They're shown in two different ways. So that's non-Mendelian genetics, incomplete dominance. There is an in-between mixture of the two, like I think red and white would make pink, or blue and red would make purple. And then complete dominance, co-shows both. So you're going to have red and white mixed together. It's not going to make pink. In this case, it's going to make red petals. Some of them will be red petals, some of them will be white petals.